thank you very much, and thank you for the organizers for inviting me this morning. So yes, clearly AI, machine learning, deep learning is starting to be used, more in drug discovery, development, healthcare. So I think it's clearly, an exciting area. So this morning, I'm going to focus mostly on the early phases of drug discovery and development, and especially on the target selection, target validation space, and I'll try to tell you why it is important. I'll have one example, very recent examples, in fact, of some work we've been doing using a learning approach, and just based on the paper was published, in fact, last week, so it's very, very new. But before I do that, I'll like, take the opportunity to maybe introduce a little bit the subject. So I think hopefully you all agree with me that, there is no shortage of data. There's a lot of data. This is growing very rapidly. Here are just some quotes that were published, I think 2 or 3 years ago in Forbes, and I'm sure some of these quotes, are still, true. The one in the middle is quite interesting in terms of, the opportunity, I suppose, so that, there's not, a lot of data is unused, and that's telling us that the opportunity is there. There's a quote on healthcare, and again, we've seen an increase of the use of advanced analytics, and I just picked up, I think it was 2 weeks ago, in the last issue of Cell, at the end of August, there was a review on what they call high-definition medicine, which is basically related to what others are calling, precision medicine, when you collate longitudinal data, multi-parameters, and it was very clear that the need to have big data capabilities goes hand to hand with precision medicine and high-definition medicine. And we all know that these changes as well are due to the acceleration and the convergence of computer science, mathematics, availability of data, and medicine. And underpinning that, what we're seeing as well, so is a lot of deals being made, and I just picked a few here. There was, the IBM Watson deal with Pfizer earlier this year. There's been a deal between GNS Healthcare and Roche and GSK as well. So we announced, that we've been working with Excensia, for example. And in the various websites, and I just picked the BBC here, they talking about AI in drug discovery and center teams when you combine machines with humans as well. So I think a lot of things are happening in the field. And I just did a quick PubMed search, last week, thinking about the talk, looking at artificial intelligence, deep learning, machine learning in titles and abstract in combination with drug discovery over the last 5 years, and you can see that that's quite a bit of increase. So clearly it looks like there is a lot more scientific activities to bring together artificial intelligence and drug discovery. Now I'm just going to say that we all know that drug discovery is a very long process, a very expensive process, divided in various phases, discovery phase and preclinical, clinical, and hopefully at the end, so you're going to have a product you can launch, And again, there's a lot of data being produced in these different phases, and that's maybe where the opportunity is, to use more advanced analytics. Now, the big issue we have, and that just some pie charts from a paper published in I think December last year in Nature Review Drug Discovery, is that the attrition. So you're much more likely to fail than succeed. So in drug discovery and development, and those pie charts are showing you in terms of clinical attrition, the cause of that attrition. And you can see the majority seems to be due. You look at phase 2, phase 3, to a lack of efficacy. So you can fail as well because of safety, or you may have some strategic or commercial reasons. But it looks like efficacy is the big cause of attrition. And that's maybe telling us that we need to pay a lot of attention in terms of picking the targets, picking the right disease, picking the right patient population as well. So. And we know, and that's a very oversimplified slide of the process, compared to the previous slide, that we make maybe 4 big decisions, when we make medicines. And the first one, which is a big one, is picking the target, so because when you've chosen the target, you start the process, and if you're wrong, so it's going to be an issue. So then you choose a molecule, then you move to human studies, and then hopefully I think you have something you can launch as well, and as a clinical benefit. And as I said earlier, so today I'm going to focus on that big first choice, which is picking the target. And why this is important? Because, and I think probably most of you know that, when you move to clinical phases, phase 2, phase 3, you spend a lot of money, so it's very expensive. And as shown on the previous slides on the pie charts, if you fail, so clearly it's going to be very costly. So if you can get better at picking the right target, so maybe you can address that issue and reduce the cost as well. So, and that's what we've been trying to do the last few years, so really trying to beef up basically the early stages, to spend more maybe around target validation, target selection, and that you see on the bottom here, and maybe reducing a little bit, the spending basically, and then trying to be more successful in the later phases. And that investment in the early phase of discovery is underpinned as well by growth of some data, which is very relevant. We feel is very relevant to that early discovery. And here is just a slide that was made at the ABI, the European Bioinformatics Institute, when they looked at the growth of data. And you can see, and you can look at the scale as well. So there is massive growth of data, both in terms of expression data, sequencing data, of course, with the genome. Proteomics is growing as well, metabolomics. So there is a lot of data that's becoming available. But what I like to point as well a little bit on the challenges we may face around data when we want to use advanced analytics. As I said, data is growing, the speed, the complexity of the data. So that's all good things, and we really want to turn that, I guess, big data wave into smart data. But I think it's important to remember that we need to ask the right question as well. So I feel that sometimes we tend to be driven by the technology, and technology, looking for a problem. And I think, sometimes we need to think first about the problem and then decide what is the best technology or analytics to use, So, and it's something that I feel it's very important. The other thing that I am concerned about, so in terms of advanced analytics, is the issue of data reproducibility. And here is just a paper I picked, that was published 2 years ago in looking at preclinical data and this issue of reproducibility. And they looked at, various papers published, some from companies, as you can see, some from academics. And I'm not sure what is, the right number in terms of, the prevalence of that issue. It seems to vary between 90 to 50%. But I really believe that that's an issue that people need to think about, when they want to use advanced analytics. And that paper, they even put a cost of that issue, so they said, there was about, just under 30 billion US dollars, and they, you can see some is due to study design, reporting, the kind of materials used as well, between different labs, So, but that's a big issue, and I think that's something people need to think about when they want to use analytics. Despite those challenges, I also feel that the opportunity is there. So we all know technology is moving very quickly. We've seen massive progress in cloud computing, cloud storage. There is progress. Look at the genomics in terms of sequencing data using NGS technologies. We've seen massive advances in gene editing using CRISPR-Cas9. We have not only genetics, we have genomics, epigenomics data. We have access to human tissues, human material. So I think there is. I also feel there is an opportunity here. So coming back to how do we pick a new target? What information is relevant? We also believe that so genetics is very relevant. We did some work on this, but others as well, like AstraZeneca, published in Nature Review Drug Discovery 3 years ago, when they shown their project in phase 2 were more likely to be successful or active if they had some human genetic information or linking that target or gene they were pursuing with the disease indication. Ourselves, we did publish, and not in Nature Review Drug Discovery, it's a mistake on that slide, it was in Nature Genetics, a paper where we shown that if you had some genetics data, OMIM, GWAS, you were maybe more likely as well to be successful at developing a drug. So we do believe that genetics evidence is important. This is not the only data we use, clearly, but it's something if you have, can give you maybe some confidence in terms of linking the gene or the pathways to the disease. And based on that, and because a lot of that genetics, genomics information is generated by the public domain, so we decided to invest in a pre-competitive consortium with the ABI and with the Sanger Institute, and that is called Open Target. And really the focus is to generate, and to select new targets and validate, some of these targets. And since the start, we've been joined with Biogen. We have also academics, that are not from the Sanger or ABI are working with us, in the UK, but also in Europe. We have some collaborators in Holland, for example, so working with us. And as I said, it is pre-competitive. All the information is shared. And so we are generating data, especially using genome scale technologies in oncology, immunoinflammation, neurosciences. And we're doing some across various diseases, So we do generate data that goes into databases, that have already a lot of data, genomics, genetics databases, Campbell, Uniprot, Ensemble. So there's Cosmic. So there's a lot of data already available. Our projects are feeding those databases as well. And what we do is we integrate all that information, all these resources into what we call the Open Targets platform. And I'm not going to talk, about the platform itself this morning, but you can use it. It is in a public domain. You can see the URL here. So, and basically, what we do is trying to integrate that data to find evidence to connect genes that are potential targets with disease. And as I said, we're using some kind of scoring system to make those connections between genes and disease, using what we call evidence strings. And we are also using some ontologies to integrate that information, for example, using the experimental factor ontologies. And here just a slide to show you the kind of data we have. It is changing, on a very regular basis. We do regular releases of the Open Targets platform, and you can see that there's some variation in terms of content. No surprises. Publications and text mining, is the largest number of associations we have. And as I said, you can find out more about the platform. There's various ways to access the data. There is an interface, which is, we believe is quite user-friendly, and you can dig into the data. And a paper, we published a paper earlier this year in Nucleic Acid Research, describing the platform itself. So what we decided to do is, we have all that data in the Open Targets platform. Of course, you can access the data through the interface, as I said, but you can access the data computationally too. So, and in terms of trying to explore a little bit that data, we decided, to see, can we maybe use that information and data we have to establish, or to try to predict, what therapeutic targets, using that information. And basically, can we, it's very similar, can we spot, it's similar to, spotting spam emails, can we spot novel therapeutic targets? And as I said, we just published that work last week in Journal of Translational Medicine. So it is very new, and I'm going to tell you a bit more about that example of using a learning approach. So I'm not going to have time to go into too many details, but I'll be available after if you want to speak about that work. So if I can change a slide, yes. Very briefly, what we done, it is a workflow here. So based on that data, so we generated a matrix. We did assign, labels. We split between training and a working set, and a prediction set. We did some exploratory data analysis, and I'll show you some results. We did test, various 4 classifiers. We did some validation. We moved to the test set, and we did some exploration, of that data, especially on the drug discovery pipeline. And then, we using what we believe was the best classifier. So we did run the prediction and did some validation going back to text mining in the literature. So it was a very very logical and very simple workflow. There's a lot of details on that slide, but I think what is important to know that, as I said, we use open targets, and the evidence was coming from 5 different features. So genetics, as I said, we feel is very important. We had somatic mutation as well, very relevant to oncology, gene expression, animal models, and pathway level evidence, because we have some pathway information. So as a positive set, we used pharma projects, which is a database that contain information about, gene targets pursued by the industry at different phases of the drug discovery pipeline, from preclinical to launch. So that was our positive data sets label, and we use, that information as well to label our open targets data. So that was our starting point. So in terms of our projects as well, so there's some caveats, so we did exclude targets that were withdrawn from the market, or where there was an notation that development was stopped. And the caveat here is that it's not always very clear if things have been stopped, because it was a bad target or it was a commercial decision or things like that. So then we used a PU semi-supervised learning approach. And the reason why we've been doing that is because, what we had from open targets in what we call the unlabeled data sets, so did contain, probably things that were really negative, but things that, could be positive as well, or yet to be discovered as positive, So that's the reason why we used that approach, So we, I said, we established a working data set, and we divided, between a training set and a test set, So, and you can see the number in terms of positives, again, coming from pharma projects, and unlabeled, So, and again, unlabeled, both negatives and things that could be potentially positive, So. We did some exploratory analysis, using unsupervised methods, So we looked at PCA, and here is a more sophisticated approach, So T-SNE that we used. And we did identify, some structure in the data that seems, to some level to distinguish, between what looks, maybe being a target and what could be a non-target, and that was done again on the working data set, So we did explore as well, as I said, we had, 5 different features, if you want, so that came from the open targets, and we did some analysis as well to see, which one were maybe most important. And you can see that, using 2 different methods here, animal model seems important, genetics again, that, that's was good in a way because it's something that, as I said, we felt was important, and RNA expression as well. So what we did, as I said, we did test, 4 different classifiers. We use random forest, we use a neural network, quite simple with a single hidden layer. We use SVM as well and use a GBM approach. So we did applied bagging as well. So because of that issue we had in a kind of PU approach. So we felt we had to do some bagging. And in terms of trying to deal with the overfitting, risk, so we use a nested cross-validation approach. So based on that, analysis, sorry, I'm trying to move the slide again. We did, I said, evaluation of the different classifiers. Here you can see an example when we use the rock curves. We did, analysis as well, AUC. We look at recall, sensitivity, specificity, precision, various aspects, So, and based on that, the neural network seems to be the best performing classifiers. But I have to say, there was not always a huge difference, So, and we had an AUC of 0.76 with accuracy of, I think, was 71%, So that's what we used for the next steps, So, and here is just we did explore the data, looking at the pharma project, the one which are in the drug discovery pipeline, and again, maybe not surprising, we found that the neural network was better at predicting targets which are launched, and was still maybe okay at phase 2, phase 3, but was not as good in earlier phases. And that we use some logistic regression as well, so to validate these findings. So after that, so we used again that neural network on our sets, that was not used for the working set, so to run the prediction. We only selected, things with a probability above 0.9, and we established a list that was over, I think it was 1,400 genes, and here just an example of that list, So, and the paper, has all the data. So, and so in terms of trying to validate, somehow. That list, and we decided to use a text mining approach. Sorry, I'm just trying to move the slide again. And what we did, we used something called SciByte to try to establish a list of genes that could be potential targets from titles and abstract. And so I think we established a list of about 4,000 as well. So. And we did look at the overlap between our prediction and what was coming from text mining. So, and we did some two-sided Fisher exact as well. So, and we did various permutations to validate the test as well. So, and you can see that there was an overlap of 590 genes. So we look at some of them, and some of them were clearly quite interesting, or we felt, where made sense, But of course, what may be more interesting is to look at the 832 that are not overlapping with the text mining. So that may be where there is quite a bit of novelty. As I said, so in terms of that piece of work, we felt that, we managed to get a decent accuracy of 71%. We did some, validation. I can put in bracket, using scientific literature. We showed that the neural network was, clearly better at predicting late-stage targets, And in terms of what were the features that were relevant, animal model, gene expression, again, genetic association seems to be the most informative one. Now, I have to say, and I think we discussed that, to quite an extent in the paper. There is a lot of caveats with that analysis. As I said, in terms of not keeping, the targets that are in pharma project that are labeled as stopped or not continued. I think that could be an issue. It could well be as well that we can really improve on that accuracy by adding more data, or maybe taking into account the druggability of the target, the genes. So that clearly there's a lot more work that can be done in that space. As I said, for us it was really we had that information data. We wanted to explore that data, and we felt that maybe using a learning approach, so was something that was interesting to us. Now, the work I've described, the open targets work, the genetics paper we published as well, so and that more recent paper on learning is has been done really by some people in the team. So I want especially to highlight Enrico Ferrero, that's done a lot of the work in the paper that I just described. Ian Dunham and Gauthier Koscielny done a lot of work on the open targets platform, and Matt Nelson work with us as well on the genetics paper. So with that, I'd like to thank you, and I'm happy to take any questions.
Okay, we've got, due to your perfect timing, we have several minutes for questions. Who would like to ask Philippe a question? If you could just state your name.
David Evans from Eli Lilly. I wondered if you in any way including safety information or descriptors that could be associated with safety or
not. That's a good question, because and because a lot of what I've described today is more about efficacy, but safety, of course, is the other side of the coin. So we haven't done it in that work, but in terms of including safety information in the open targets platform, yes, we're working on that as well. So absolutely. Okay. So. Yeah, because it's interesting, the open targets platform, we initially we did it mostly for, the idea was to help biologists to look at that information in terms of target selection and target identification. But in fact, a lot of open targets have been used as much by people in safety at GSK, because we have information on animal models, or we have some phylogenetic analysis as well, various things that people are using. So yeah.
Great, thank you.
So next, who went next one?
Hi, Philippe. Joanna Holbrook. I was wondering, do you see improved accuracy for particular therapeutic classes? So if you segregate by disease?
No, we're working on that, so because that's one question, so we do know, I cannot, we've been trying to investigate, in the context of that learning. I think one issue we had is the volume of data. When you start to subdivide by therapy areas, if you want, the data is not as big, so, and I think that affects, the way I think the approach we have, so. What we do know is, because we've done a bit more work specifically around, one data types, the genetics, so we do see some variation, so in the different therapy areas, on the genetics.
I assume it depends on how many positive cases you have as well.
It depends how many positive cases, I think, absolutely.
So you're better for cancer?
Cancer is not bad. Well, yeah, I mean, yeah.
Can I ask a question?
Yeah.
Philip? How are you then integrating this with your target selection for programs that you work on within GSK?
How do we? So in terms of the open targets, as I said, we do have that evidence, and we score that evidence, and we have something called QBA, question-based assessment, or something like that. So basically we have, and it's an electronic version. So we do integrate that information in these assessments of targets. So that's part of a. We have something called a target TQQ, target quality questionnaire, and that's part of it. So.
Okay.
Hi, Savi Stel. Do you you mentioned duplicity of data and quality of data. Moving forward, do you have any any ideas on? I mean, do you have the capacity and the power to implement newer strategies to improve the quality of data? Any technologies? Any specific?
That's an excellent question. So the problem is, a lot of data we use that has been published, a while back as well. So, and that's what I don't think I've got a good answer. I think because I know that at GSK, so sometimes we see publications, we try to repeat the experiment, and that's not exactly, the result is not exactly the same. So, and that's a big issue, I think. So.
We've got time for one more last question.
Hi.
So.
Guy Moss from UCL. I have just related, following on from that last question. Scientists are very much aware of their, things like their H index. Should we have an R index, a reproducibility index? Should each scientist, should their output of their lab be ranked for reproducibility as well as, the impact factor of the journal that they're publishing in?
That's yes, why not? Absolutely, yeah. I think that's a good, yeah.
On that
good
positive
point.
note, I think
positive
we'll
note.
move
Yeah.
to the
I'm
next.
not sure. How everyone will react to it, but yes, it'll be.
Okay, I'd like to thank Philippe again. Thank you for a great talk.